Brilliant. Thank you very much. Great. I'm absolutely delighted to be here. So many, many thanks to Jeremy and the ProImmune team for the kind invite. I adore Boston. In my previous life, I was here on a number of occasions, and it's a fantastic hub for life sciences. And so it gives me a great opportunity to share with you the work that we're doing on these unique domains. And as you've gathered from the first slide, we don't work on human platforms, and indeed we don't even work on mammalian platforms. The platform I'm going to tell you about has been in existence for some 450 million years. So I think questions of immunogenicity always arise. So this gives me a great opportunity to get some feedback from some experts in the field, from you guys. And what I'm going to tell you about are these VNAR domains. They, of course, are isolated from sharks or Elasmogen. And the reason I'm telling you that is to give some rationale behind the name of the new company that we're starting, Elasmogen, which will be based on this VNAR platform. So you may be able to tell from the accent that I'm not local. I come from Scotland, the top right-hand corner of Scotland in Aberdeen, and this is where we're operating from. And there is a small cluster of biotech companies there which focus on biologics, particularly antibodies and peptides. The position we're in right now is that we've managed to put together about $3 million of pre-commercialization funding in order to develop this platform further and then spin out this company called Elasmogen. We're basing ourselves very much on these VNAR domains, and we're utilizing the USPs that they will deliver, and I'll give you a little bit of information about them as I go through the talk. We have a pipeline at present based on a biotool and a number of therapeutics. What I'm going to do today is I'm going to focus on one particular biotool because that's the one we've taken furthest through, and that's the one we're actually looking at potential immunogenicity questions with. We have a strong IP portfolio, so the manner in which we're doing this is that we're protecting basically the platforms that we operate off of, the libraries that we're creating, and the products that we've got coming out of those libraries. And I'm delighted to say that we have a fantastic experienced management and science team. Many of the science team are ex-biotech, ex-pharma, so although I'm operating out of the University of Aberdeen, we really are operating at the level of a biotech company already. So this is what we're all about. We're all about taking these unique domains, these VNAR domains, from these beautiful animals and developing them towards the clinic. So what are these domains? What's the deal? VNARs form part of the so-called immunoglobulin new antigen receptors. These are part of the adaptive immune system of sharks, and this is what they look like in the shark blood. So they're homodimers, and what you'll notice first of all is that they're single chain. They don't have any light chains. They have about 5 constant domains, but this is the part that we're interested in. This is the variable binding domain. This is the business part of the molecule, if you like. And this is the part of the molecule that interacts specifically with high affinity to target. And it's completely amenable to phage display. So this is the part that we're isolating and using as our platform. They can boast the fact that they're the smallest antigen binding domain in the animal kingdom. They're inherently soluble and stable. And what's interesting, and you will note as antibody experts, that they differ very, very slightly from classical antibodies. They have a CDR1, they have a CDR3, but they have no CDR2. There has been a truncation event, and they don't have any CDR2. And because that's the region where it would have interacted with its partner chain, the residues in that region are now quite hydrophilic in these molecules. So although we've lost one region of diversity, we have gained 2. HV2 and HV4, which are based on the TCR nomenclature. And what we've got with these domains is we have a number of different structures that come out of the different isotypes from different families that we've got. And they differ with the number of non-canonical cysteine residues within these domains. And what that translates into, essentially, are different structural families. So this is a USP about these type of domains, is that we don't only have one type of structure, but we have multiple types of structure. So here, and they're very interestingly just called type 1, type 2, and type 2b. So not very interesting names, but very interesting structures. And what you can see here is the addition of non-canonical cysteine residues across the CDR3 and across the framework residues in this particular type composes a constrained, very stable type structure. Here, in the type 2, we have an extra cysteine bridge between CDR1 and CDR3, and that results in a protruding type structure. And then we have this third main type, structural type, which lacks these extra non-canonical cysteine residues, and it's completely flexible. So we have lots of variation within these structural families. And what's quite interesting, particularly with the type 1 and type 2, is these extensions of the CDR regions are conducive to binding pockets or canyons within their target molecules. So both the type 1, which is more constrained, has the capacity to do that, but very much so the type 2 can actually stretch right into perhaps considerably more cryptic epitopes, if you like, more hidden epitopes. And yes, size matters, but not in the way you may think. So in our world, small is good. So everything that we want is compacted into a 12 kilodalton binding domain. And all I'm showing here, just to give you some context, is a classical antibody, of course, coming in at 150 kilodaltons, and progressively smaller development that's happened in the field towards smaller binding domains. These are the nanobodies, so these are the single chain domains that come out of Camelid, and this is the formulation of one that's in the clinic at the moment in this trimer formation. We are arguably about 10 to 20% slightly smaller than the nanobody. We operate off of two platforms, immunization. So we do, in fact, immunize sharks. And the beauty of doing that, as with any animal, is that the animal does the work for you. So we inject the immunogen, and the animal will go through a process of in vivo maturation and produce high affinity VNAR domains against that target. Another positive about this methodology is that they are, of course, evolutionarily distinct from us. So any proteins that might be highly conserved between mammals, it may be that a shark has never seen it before. And one of the examples I'll give you today is about one of our lead domains, which is against albumin. Sharks don't have albumin, so that was quite an easy target to use. But of course, what we've learned from these VNAR domains is a lot about the sequence and the structure, and we can now extract what nature does and recreate it, of course, in the laboratory. And we have a number of synthetic libraries that we've built, and what we can do is we can interrogate them against a number of different targets and pull out therapeutically relevant VNAR domains from those. And they're delivering quite well at the moment. We're getting low-digit nanomolar hits from these libraries, which is quite unusual from a synthetic base. So just to bring this all together, to give you a bit of a kind of highlight about why we like VNARs and why we're working on VNARs, is they have inherent properties. So they're high affinity, that's their day job, they're part of the adaptive immune system. Very small, very soluble, and very stable. So from a manufacturing perspective, that's very attractive. They don't have any glycosylation sites on them, and so we can actually express them both in mammalian systems and other prokaryotic systems. We operate off of two platforms, and because they're single chains, it's very easy from a molecular biological perspective to make different types of formats, very simply to do that in the lab. So we can create bispecifics or multispecifics. And we feel that they're perfectly adapted for ADC development, which is something that we're progressing at the moment. But I think a big USP for these, compared to the other single domains that are out there, is this natural existence of different structural families. So we have flexibility in the structure, but also a lot of diversity within these domains as well. So let me take you through our lead domain program. This is, as I mentioned before, an anti-albumin binding domain. It came from an immunized shark. It was immunized with human serum albumin. The domain is called EO6. We isolated it through phage display, and it has picomolar affinity. We now have a crystal structure, which was very informative for the process that I'll tell you about in our humanization strategy. It's been through a number of different animal models, both rodent and NHP. And so far, but I'm scared to say this in front of the company that I'm in, the immunogenicity doesn't look as if it's a problem, but we're addressing that, we're looking at that, and that's why I'm here today to talk to you about it. Now, the purpose of EO6 is not as a therapeutic in its own right. It doesn't really matter if you target albumin. The purpose of this particular domain is as a PK tool to enhance the half-life, the serum half-life of components that are fused to EO6, if you like. So the idea there is something that's been used before for PK enhancement. You bind to albumin. Albumin has a long systemic half-life, and so you increase the half-life of anything that you fuse to EO6. So I'm putting up one slide, but a lot of work went into this domain to make sure that it stays stable and binds HSA at high pH and low pH. It doesn't interfere with FcRn binding. All of these kind of things were all included into the selection process. So we had to find a partner for EO6. We didn't want to put EO6 into any PK models on its own. What we wanted to do was to create what it would look like as a product. So it was always going to be N-terminally or C-terminally or both ways fused. And what was also important was for us to understand whether or not EO6 would tolerate being tethered at the N-terminus and the C-terminus. And the answer to that is yes. The partner domain that we chose to fuse to EO6 is something called 2V. 2V is another VNAR domain, but it binds, as far as we're aware, nothing. It's a dummy or a naive VNAR domain. It came out of the original sequencing database that we put together when we were generating domains for the synthetic library. So what we're asking questions of here really is EO6. We're not so bothered about the cognate partner at the moment. But when we looked at the affinity, for example, when we make the variations with EO6 on its own or fused to 2V in various formats, we can see that we have high affinity for human, we retain high affinity for mouse and rat, and also monkey. So we get cross-species reactivity, which is not really surprising because albumin is quite highly conserved across these species. But what's nice is it does tolerate, and we get retention of high affinity when we do fuse it in various formats. But the key question is, does it increase the half-life of a fused domain? And the answer to that is yes. So this is an early piece of work that was done. This is done with iodinated material. We're looking at the PK profile in mice. And here you can see that if you inject 2V on its own, you get a very rapid clearance from the plasma. Whereas if we put in a fusion of EO6 and 2V, we get an extension of about 48 hours. So it looks like EO6 rather is doing what we hoped it would do and increase the half-life of 2V. We then stepped it up and we looked at non-human primate models. And here what we're doing is we're looking at two variations on a theme. We're looking at an N-terminally and C-terminally fused version of EO6 and 2V. And these are IV injections here in the red and the blue. And you can see that when we inject that, we get a nice straight linear PK profile, and we get a half-life of almost a week in these animals. And what we then did was we took this particular domain and we went back into the same animals again, but this time subcutaneously, as shown in the black line here, and we get rapid bioavailability into the blood, and the profile basically follows the same line as we saw with the IV. So we were really pleased with this because this provided evidence that these domains can go in through two routes of administration, and we're getting extension of half-life through both. But also what we didn't see is that because this is a repeated dose, we didn't see any more rapid clearance, which may be indicative of something like ADA being produced in these animals, binding the complex and clearing it more rapidly. So this provided us a little bit of confidence that there wasn't anything untoward happening in monkeys in this particular model. So what we've done actually is we've gone through 3 different PK models in mouse, in rat, and also in cynomolgus monkeys. And what we're showing here really is the linear relationship between the half-life of albumin, which is shown in blue here. So this is from published data. There is a linear relationship between the size of the animal and the half-life of albumin. And what we're doing here is we're plotting our actual data on these points. And so what we are confident in saying is that we can extrapolate from the data that we've achieved to date and be predictive of a half-life in humans of about 19 days. So we set out to produce a VNAR PK tool that will bind albumin and extend half-life. We've produced that essentially. And that's a key biotool not only for our internal development, because if we're working on domains therapeutically that are only 12 kilodaltons, they're going to be cleared very rapidly. We have to have a capability of extending the half-life of those. So this was a great achievement for us, and we were really pleased with this. Now what we've also done is we've done a level of in silico analysis of E06. So during the process of developing these domains, I always get asked two questions. The first question I get asked is, how do you immunize a shark? And the answer to that is very carefully. And the second question is, okay, Caroline, these are coming out of sharks, are they immunogenic? And that's a question that's really important, right? I mean, we're all sitting here today discussing this. And so this is why we're going through this process with this particular lead domain, to understand these domains a bit better for the realms of immunogenicity. So we've done some in silico analysis, and what we found here using this particular methodology is there's no big red flag waving at this point. So E06 comes in round about where IgG-Fc region is and below albumin itself in this particular analysis. So we then entered into an immunogenicity analysis of E06 because it was our lead domain and we really wanted to use this as our model domain to understand this process a bit better. So the question is, are they going to be problematic, these domains? Can we de-risk E06 and so it's ready for partnering and phase 1 development, and also to provide invaluable data for the future development of these VNAR domains? What do we need to do to make them less immunogenic, or are they not immunogenic in the first place? Should we be bothered or worried at all? We went through a humanization process using E06 as our model, and there are a number of different ways that you can do this. Now what's interesting about VNAR domains is if you look at them from a primary sequence perspective, they're not similar to IgGs, okay? They've obviously come from some other lineage, and we're right down there at about 25–30% sequence identity. However, if you look at the actual structure of them compared to human frameworks, they're really very similar. So that was kind of the methodology that we adopted, was finding which human framework was most similar from a structural perspective, and then going through the process of resurfacing that. And what's really important during this process is that we don't want to ruin the VNARs, right? The VNARs are single chain, they're soluble, they have a number of attributes in there, so we have to retain hallmark residues, regions that are core to those functional abilities, and change the external, if you like. That's the approach that we took. And this really is just to illustrate in block colour, to make it clear, because you have no hope of seeing sequence sitting in an audience, is really to understand the iterative process that we went through. We chose DPK9 as something that was a framework that was structurally similar and also behaves well from a biophysical perspective. And the first domain that we did this with was actually a 5A7 domain, which is an anti-Hennig lysozyme VNAR domain. And we went through the humanization process, the resurfacing process with this domain, and we showed that we managed to retain binding and functionality of this domain. So we used this as a kind of template for humanizing E06. And we started off, as you can see here. So sorry, I should have made this clear. The green is human, the black is shark, and the red are CDRs. Forgive me, I should have said that at the start. So this obviously is totally human. The 5A7 started off as obviously shark, and then that's the level of humanization that we did with that one. This is E06 with the level of already retained human or existing human amino acids in there, and we went through an iterative process of changing amino acids to make them more human. And during this process, of course, we check the functionality of the domain as we go through, because that's key, is to make sure you've got something coming out the other end that actually works. Now during this process, we were very fortunate to get the crystal structure of E06 with albumin, and this was incredibly informative. So we can now begin to understand what the interface residues are between the VNAR domain and its target. And this particular VNAR domain falls into that third structural family that I showed up earlier. It's a flexible VNAR domain, and the interface is quite interesting. It's quite planar for a VNAR. So what we can then do is we can look at where those interface residues are across the native molecule, mainly revolving around the CDR3, but we also saw regions in framework 2, HV2, and a little bit of CDR1. And what we could then do with our humanized iterative domains is we could mutate some of those amino acids back to the interface domains to get a better binding molecule. And so what we've ended up with, to cut a long story short, are a number of lead humanized E06 domains that retain functionality. And this really is just to give you an illustration of what those domains now look like. So here in the purple are the original Shark amino acids, and this is the interface that was important to retain for the functionality. And here you can see that we've gone through the resurfacing process, and these are the human versions of that. So this is human, whereas this is Shark. And again, we've done a little bit of in silico analysis on where these humanized EO6 domains lie, and they're very similar actually to the native. So we haven't gone anywhere up the ranking or particularly down the ranking. It's all pretty safe with regards to this in silico analysis. So where are we right now with this particular domain? Well, as I said before, we have a number of these lead domains which have gone through this humanization process. And this is the world that I'm in right now, which is what we're all talking about, which is wonderful. So I've heard a number of people talking about the fact that these assays are incredibly good to do some ranking of your humanized domains, and that's exactly what we're doing, because we're looking to pull a candidate out of this group of humanized domains that we've got. So we're currently carrying out DCT assays, and I guess if we see nothing or little or whatever that readout may mean, relative to the controls that are in there, we may consider expanding those assays just to confirm that, and from that we can possibly pull out a candidate human EO6. But if we see something there that looks untowards in these DCT assays, then what we might try and do is hone down on the sequence, so looking at the in silico, which will inform us about that, design some overlapping peptides and see if we can decide where that hotspot is, if you like, if there are any hotspots, and identify those. And then, of course, go through a process of re-engineering, which is the beauty of what we can do, is we can change to a certain degree a number of those residues to de-risk that, and then go back through the process again and make sure that what we've got is okay. So we're actually in these assays right now. So I can't tell you the result of them. I can come back in a couple of weeks and tell you what the result is, but we're just waiting. So just to conclude, what I've told you about is probably something quite different from the kind of domains we've been talking about at this conference. So we're firmly in the world of Shark VNAR domains. I've talked about EO6, which is an incredibly effective tool. It works really, really well. So far, the work that we've done didn't suggest anything horrific going on from an immunogenicity perspective, but we didn't really interrogate it properly, and that's what we're doing at the moment. So we have gone through a process of humanization, because we feel that no matter who we speak to, they're still going to ask us whether or not we've humanized it or not. So we've just basically taken that up front and said, early stage development, yes, we have. So we're taking the native and the humanized through these assays to compare them. And essentially, I'm really excited, because regardless, this data set will inform us. It will either de-risk EO6 as a domain that we can take forward, or it will provide us with a humanized version of that. But all of that data set will provide us with really valuable information about our next series of VNAR domains that are coming through our pipeline. And I could not go away without thanking the fabulous team that I now have in Aberdeen. John Stephen, who's here today, Marina, Laura, Gerard, Whaley and Obina, who are all working on these domains. And of course, I wouldn't be here if we didn't have funding to continue with this work. So a lot of this has been funded by Scottish Enterprise. We've also been awarded a follow-on fund from the BBSRC, and I've continued to get great support from the University of Aberdeen. So that's it. Thank you.
Great, thank you very much, Caroline, for introducing us to the world of Shark.
I know.
I'm sure we've got a few questions. Anyone else start off?
Oh.
Yeah, I've got a question. You could also.
I was just wondering how similar are these ones to the Camelids
Yeah.
molecules that has been around for a while, and what do you know about the immunogenicity from those ones? They must have been in the humans by now.
Yes, there are. So, I mean, the Camelids, or the nanobodies, have been around for quite a while now. And I think they've got 6 clinical programs ongoing at the moment. The company is Ablinks, of course, which is in Belgium, beautiful Belgium. And the answer to your question is they're really quite different. So it's obvious from an antibody sequence and looking at them that they've come from an IgG lineage. So they still have a CDR2. They've gone through some kind of process where they've lost their cognate chain, their partner chain, if you like. So the similarity to sequence level is something up at 85% already. But I do know that they do go through a level of humanization before they go into the clinic. I couldn't tell you if they've seen any issues. I haven't read about any and I haven't seen any. But with the sharks, although they're also single chain, what's happened basically is it's an example of convergent evolution, that an animal in the desert and an animal in the ocean have come to the same conclusion with respect to adaptive immunity. But the sharks, as I said already, the sequence is different. So it looks like, from an evolutionary perspective, they've come from some other member of the immunoglobulin superfamily, perhaps ICAM or VCAM or some cell surface molecule. So they are quite different. They're the same but different, a bit like us. Yeah.
So when you then humanize these molecules, do you find it harder to retain the affinity compared to, let's say, humanizing normal IgGs?
For this particular domain, I mean, we've actually got affinity data, so I mean, I've not really gone through all the checks that we did on these humanized domains, but we went through an iterative process. But what we managed to do at the start of the process was to actually select based on affinity. So we made changes. We started with a template, a humanized template, and made alterations to that, and then actually used the ability of phage display to pull things out based on affinity. But yes, it is a critical factor when it comes to retaining all the attributes. You know, it's a case of, we like the VNARs for certain reasons. We don't want to lose any of that when we humanize it. So it's that balance with how much do we need to humanize and change this. That's always the case. And what's quite interesting is when we started this process, the choice was to either start with a shark domain and humanize it, or consider it as starting with a shark domain and to sharkinize or to humanize. So to kind of come into the middle at that point and get something that works from both perspectives, I guess, yeah.
Yes, thank you. That was a very interesting presentation of the VNAR. And I had two quick questions. One, is your PK assay measuring the intact molecule, or is it, I know it's a two-domain, or is it only looking at EO6, for example?
That's a very good question, because that work was done by a member of the audience here, a beautiful piece of work, and we're looking at both the domains. So it's LC mass spec that we're looking at here. So the first piece of data that I showed you was iodinating the EO6 2V, and you're right, we don't know what comes out the other end, to be perfectly honest. We don't know what we're following. So a lot of work went on to optimize an LC mass spec methodology. So we've got signature peptides across EO6 and 2V, and we're following the actual two components as we go through the whole process. And I really cut through that data very quickly. I mean, an awful lot of work went into that, and we also have PK data to support the trimer as well, so the EO6 with two domains attached to it, and it works perfectly well as well.
Great, thanks. And then the second question, was there any concern, or did you have any interpretation of the fact that there was no change in the in silico result when you went through such a drastic transition?
Yeah, I know. I mean, I guess, I'm basically looking at this from a learning perspective, and we went in and did the in silico. We had no idea what we would see with the native domains, and they really don't look like there's any big problem already. So what we're essentially doing, in very simplistic terms, is taking something that doesn't really look like this big problem, but putting human sequences in there, which we would hope wouldn't have a big problem anyway. So we're not really seeing any change from the in silico. And my understanding of the in silico is it's a great tool as a start point to bring up anything that could possibly be a problem. So I mean, there are areas, I'm not putting my hand in my heart and saying it's completely under the radar. So it may be that when we get more information from the subsequent in vitro assays that we've got, that we can utilize that information to go back. But yeah, there really are. There's very little difference between the native and what we've essentially humanized. Yeah.
Okay, I know we spoke a little bit about the immunogenicity related to, I guess, the structural stability of a particular domain, with the idea being that if you've got a lot of stability, maybe it's more difficult to go through the processing step. Of the molecules that you're putting into the DCT cell assay, do you have kind of a range of structural stability? I think you probably measure thermal stability, do you?
Yeah, no, you're right, actually. So it might be that, that if we can't break it apart inside, we're not going to see anything coming out. Yeah, we've got some biophysical characterization across the domains. We don't know if they differ hugely with respect to stability. They're stable anyway, but I don't think there's a massive variation between the humanized and the native domains as per se. But that is a factor we've got to think about. If we don't see anything, is that because of the processing and the stability of them inherently, or what the deal is, We're building up a picture of all of these domains at the moment, I think, is probably what I'm trying to say. So we've already looked at, I know we talked a lot today and yesterday about aggregation, so we looked at that, for example, to see if there was any inherent aggregation. We've done a level of stability, but there's not a huge amount of difference, and of course the affinity and so on and so forth with these. And I think what we're trying to do here with the DCT assays as well is have a funnel, so we can feed all of these things into it and have a candidate coming out. But yeah, absolutely, it's this consideration that we have included, but I don't have time to show all the data. Okay.